It gives me great pleasure to welcome Dr. Gerd Binnig, former Nobel Laureate. I shouldn't say former, it's always Nobel Laureate. Sorry, my bad. Yeah, in physics, he's been gracing the presence at Clinam for the last several years, and he's going to talk about his company, Definiens, and I think they do some of the image analysis and that type of work, which I think there was a question, how do you interface with the patient, and does the computer read the patient from the time he comes in? But maybe he has some answers for that. Thank you very much.
So good morning, and thank you very much for the opportunity to give a presentation on our work here. It's not the first time that I'm here. I think it's the third time now, and I always enjoyed it very much. The title of my talk I have actually stolen from you, from IBM. It's very much related, but it's on the other side of the world. So on one side you have all the texts, on the other side you have all the images. And I quote here IBM also, in the internet you can read 80% of the data in medicine are unstructured, but they are relevant. And that's why automatic structuring is required, that you can make sense out of this information. And IBM uses the term cognitive computing because you bring in some of the capabilities humans have, how they think about problems, and simulate that. And we actually try to do something very similar and do the automatic structuring this way. The automatic structuring might be done on the fly. So we actually go a step further by saying it's more than 80% that's unstructured. Or you could say that's unavailable because it's sometimes even not digitized. So it's not available for a computer. And a good example for this are tissue slides. In particular in oncology, all the important decisions are made on the basis of tissue slides. So people just take those tissue slides and look at those slides under the microscope, and something like 100 million slides are produced every year. And those, if you take them into images, if you scan them then, and with a resolution that's available today, each of them has something like a gigabyte. So if you sum that up, you have something like 100 petabyte per year that's produced in data, but it's more or less lost because it's not digitized. And the pathologist just looks at those images, at part of those slides, and comes to a conclusion. Is it benign, malignant, or what kind of treatment do I need to apply here? And they all know, they just have a quick look at those slides, that there's much more information in those slides than they can take out. And that's where the computer can help a lot. And unfortunately, the world is changing right now, and those slides get more and more digitized by using those scanners, where you can scan a slide relatively fast, in, let's say, a minute or a little bit more, and can look at them on the computer screen. And once you have that, you can actually automatically structure those data, because in principle, if you just have a bunch of pixels, that's unstructured data. You can't do anything with it. You cannot bring pixels into a data mining tool. You wouldn't know what to do with it. So here again, you need to automatically structure those data, and you could call it also cognitive computing, because it's by far not trivial what you have to do, and we call it cognition network technology, how we do it, how we structure those data and come to some conclusions. You can call it datafication, image analysis, and at the same time you quantify a lot. So in principle what you can do, you just group pixels into meaningful objects. This way you structure those images. And then, once you find them, you associate those objects with a meaning, like nucleus, for instance, or gland, and then you can characterize them and quantify them in many different forms. And then you can use it for big data. Yeah. If you do that on a large scale, and the time is right now because all the components are there. Now you have the scanners. You can digitize all these slides in a relatively short time. The computing power and the storage is available and is not too expensive. Systems biology is there already at a certain level where you can use it to come up with hypotheses, what to look for, and all the tools for cognitive computing, not all the tools, but most of the tools are already there, like machine learning and other techniques. So we call that, if you apply cognitive computing to tissue slides, we call this tissue phenomics. And it's somewhere between the molecules on one side and the patient on the other side. Because what you want to do is we want to correlate the data we get out with the clinical outcome data, the clinical data in general. So then we bridge the gap between the molecule and the patient. So what you in principle do, you do the datafication of all those slides. You take out all the information you can grab, and then you put them in data mining tools and look for patterns. Correlate all the data you get out with clinical outcome, for instance, and with other clinical data. And then you can ask the question, can you find patterns in those images that predict something? For instance, drug response, in case you want to treat the patient, or the probability for cancer recurrence, so prognosis. Can you predict that just by looking at tissue slides, or the machine looking at tissue slides? And you definitely can, because people do that all the time. Pathologists do that, and they make treatment decisions when they look at the tissue slides. But if you do it in a more quantified way and do the big data approach, where you even find new types of structures which pathologists have not discovered yet, you'll bring everything on a much higher level. And the predictions pathologists make today are not extremely precise. They know that, and everyone knows that there are a lot of errors in the system, and hopefully they can be reduced. So pathologists look at these phenotypes by looking at those structures, and from these phenotypes, including also kind of genetic data in space, like by in situ hybridization, and looking at certain proteins that tell you something about the functions locally in these images, they come to the conclusions. So the next step would be just to take what they know and look for those objects and quantify them. Just quantifying them, get a lot of numbers out there, brings it already on a new level. But then through big data approaches, you can also do it in a way that you do it more or less hypotheses free, and just look for new structures you haven't discovered before. So once you do this quantification, we call the structures that are relevant in terms of that they mean something for the clinical outcome. We call them phenes, in contrast to phenotypes, what the pathologists are looking for. We call them phenes related to genes. And if you discover new types of structures, we call them novel phenes. And we have already discovered a few years ago that if you just look at nuclei and quantify them, you can predict something. Yeah? You can divide groups, patients into groups, long-living, disease-free survival time, and short disease-free survival time, and you get a relatively good prediction and a good p-value, high probability that these results are right in a twofold cross-validation. And in the meantime, quite a few groups are working on that and getting very interesting results. Discover completely new phenes, like Jérôme Gallon in Paris. They work also with our technology, and developed this immunoscore, which is in principle a novel theme. Yeah? Just in the microenvironment of a tumor, you look for the inflammatory cells and measure the certain ratios of certain cells you discover there and quantify that, and you come up with a new score which is very, very predictive, great results. And in other places, like in Edinburgh, Peter Kay and David Harrison, they worked on colon tumor where budding of cancer nuclei in little clusters have a very high predictive value, and if you quantify that, you also come to conclusions a pathologist would never come to. And Andy Beck, for instance, in Harvard discovered novel phene, which is just looking at the structure of stroma. Pathologists would not look at. He just looks at the tumor region, not at the environment. And he found also here quite a predictive value in those structures of the stroma, and so on. And more people working on this theme. And we want to do that now on a little bit more well-defined basis by bringing different types of information into one image. So you have the morphology, you have all the different stains. Let's say H&E is the basic stain people use today to just look at the morphology of the structures. But you can combine them also with IHC, marking certain proteins and functions, or even ISH by in situ hybridization, and bring all these information into one image. Can do it in different forms by multiplexing with fluorescent imaging or washing. There are several techniques how to do this. We do it just by co-registration of those images, bringing them just align them and bring them all into one image. And what you probably want to use today is immune cell markers, because we know they have predictive value. To make image analysis a little simpler, you might want to mark epithelial cells or basal membrane markers might be used, proliferation markers pathologists use anyhow, and all kinds of standard markers that are already used in pathology, and maybe also in the future more and more nanomarkers. And bring that all into one machine. And then we copy more or less what's already done in genomics. So you collect all the contents from the clinics and bring it into a big content base, database, with clinical outcome of the patients and all the different images that were produced. Bring that together and try to datafy, and then do the image mining on top of that. And once you discover that, you might discover relevant structures which you then call phenes. And from those phenes, if you combine in a certain way, you might create tests like that's done in genomics for the different types of organs. And once you found those structures and you can predict something, you can apply it in the clinic for an individual patient. And then you have a portal to do that. So the reality is a little bit more complicated. It's not just collecting data, doing all the image analysis, then do the data mining and finding relevant structures there. It's a little bit more sophisticated because you don't actually know exactly what you are looking for. So datafication is a little bit more than just image analysis. And what is here very helpful are heat maps. So you can plug in heat maps to reduce the amount of information that's in these images. Certain features, for instance, in red, it's a tumor region, or you can say a certain density of mitosis or whatever, or certain stains, density of certain markers in a certain region gives you a hint. But it's not so clear what you actually want to display. Here in this case, it gets more and more common to use heat maps also for other purposes. It's just that these days, I mean, that's used just to display where the players in the soccer team are, but it's not so absolutely clear. What do you learn from this? So what do you want to display? What kind of features do you want to display on such a heat map? And what kind of structures are actually relevant that tells you something about how the game went? And it's the same for tissue slides. So what we actually do, we measure from all these different images all kinds of features, and we display those results as heat maps and bring those heat maps into one heat map. And then it's not so clear what to look for. So if you don't know what kind of phene you might want to discover, then you have to play a little bit with the system. So you start with all the different patients, with all the different stained images in the beginning. And you do an initial image analysis on that, and you export some kind of results. And in principle, you can export everything you want in terms of heat maps, where you display certain features. And you bring all these heat maps into one image by co-registration for each patient. And then you have a new type of image, a heat map image, a complex, we call it hyperspectral heat map, and do, again, image analysis. But you don't know exactly how to do the image analysis here because you don't know what kind of objects you are looking for. But you do an initial segmentation of that. Then you extract certain features from objects you found in these heat maps, put them in a data mining tool, correlate those results with clinical outcome data, and you get a result. The result is either good or bad, but you store the quality of the result and the features and the parameters you use for achieving this result you store at the same time, and put everything in a database, and then you reconfigure. You play a little bit with all these parameters, and in particular you can go back to the heat map because there it's most unclear what you're looking for, and just segment a little differently, export some features a little differently, and with a new configuration, and you again get a quality. It might be better or worse than the one you got before, and you do that many times. The heat maps are relatively small images. You can do that in a very fast fashion, and you remember all the results. In the end, you get one of them is the best one, and the best one might not be the one you use because you want to have it also very stable. Yeah. So if you just tweak the parameters a tiny little bit and the result gets worse, then it's not a very stable result. So the stability and the quality that you get out is important. And this you have to do also in a more sophisticated and intelligent way to go in this loop to come to a conclusion. And we use the same technique, this context-driven technique, as we do for the image analysis, we do for this loop. So that then you start in the beginning with a huge set of stains, and you don't know which stain is actually a good one that has predictive or prognostic value. And you run the machine and you reduce the number of possibilities. In the beginning you have a huge amount of possibilities, what kind of objects you want to look for and what kind of stains you might be important. And in the end you boil it down and maybe to only a few of the stains which are really relevant, and to a few objects which are relevant. And then you found a new phene, or maybe several phenes, and you can build a test on top of that. So the immunoscore, which was developed by Jérôme Gallon and his colleagues, would have been invented automatically if you do it this way. If you have thought about that you want to mark immune cells or the different immune cells, if you have thought about this, to put that into your stain collection, then you could have invented it automatically. And more than that, you can optimize it automatically because it's not absolutely clear the what is the tumor microenvironment, this invasion front, big or small. You can model this, shape it bigger or smaller, and see whether the quality you get with respect to correlation to clinical outcome data gets better or worse. And this way you can optimize that. So that's my last slide. Just have 20 seconds left. So that's just a last idea how to connect everything and connect also to Watson. So we do the imaging and we do try to automatically understand images. And we also, this way we also have an ontology where we find certain objects in a class hierarchy. They are related to a class hierarchy. I find nuclei. Perhaps I have a term like triple negative breast carcinoma in a certain region. I found that. And Watson has also an ontology, and maybe has exactly the same term, and maybe a combination of different terms like that. And then it connects to Watson. And then Watson might come to the conclusion, because he has this more generic worldwide knowledge, that in such a case where you have a certain combination of features in an image, that you want to perhaps in a certain region make some gene analysis, or picking certain regions in the tissue and do gene sequencing here and come to a more comprehensive conclusion. So it's kind of a clinical decision support system. Watson might be in this case proposing a new experiment or a new test that comes on top of it, and in the end, bringing all this information together, you might come to a much better and more comprehensive conclusion how to treat the patient. So with this I want to conclude. Thank you very much.
Thank you for a very interesting talk. Is it open for questions?
I look at a lot of slides, and there's three things that come to my mind. One is artifact versus non-artifact, and the propagation of errors due to artifact. The second thing is, as you know, when a pathologist is doing this over and over again, a pathologist that has been doing it for a while does a better job in some cases than other pathologists. And are you organizing the data in such a way that it is taking that into consideration, where you have a good pathologist or a pathologist that really knows what they're doing to categorize it, and is that getting a higher rank versus the pathologist that may just be starting out?
Yeah, yeah.
And then the third part of that question is, going back to the doctor, how quickly the pathological tissue is being done, and when is it being done? Is it being done interoperatively, or is it being done post-operatively? Because the preparation is going to be a little bit different, which will generate more artifact. I mean, these may be unfair questions, but these are issues that you run into all the time.
No, you are perfectly correct. The biggest problem actually in image analysis are the artifacts, because they are most difficult to recognize, and we have to fight with those. And I have the impression we get it under control, but it's definitely the biggest challenge. Just finding the nuclei, things like that, it's relatively straightforward. But the artifacts are sometimes could be very, very different. But I have the impression we get it under control. At least we can give at the end a quality factor out that tells you, here we are not sure, here it looks like there are a lot of artifacts, please look at it, or rescan, redo the slide again. Yeah. So that's certainly a problem. And the other problem, you are also right, the annotation. So if you use the ground truths from the pathologist, we experience big problems, because we hand over one on the same slide to several pathologists, we get very different answers. That's how it is. So for us, this is only one component to get the feedback from the pathologist. The other component is the clinical outcome data, which is also a kind of a ground truth. And maybe sometimes, if you do it right, more valuable ground truths than just annotations made by the pathologists. Or the combination is very extremely valuable. So then you filter a little bit out bad annotations. Yeah. We have here interesting experience actually. Yeah.
Question over here.
Thank you very much. And I just want to ask, following what was asked right now, if you take those images and you do kind of, as you said, cluster or patterns, as you called it, unsupervised, without the pathology, before he's telling you anything, just look on, as you said, on the big data, on the broad images, and try to cluster them according to the features or according to any other analysis that you are doing. So that's one question. That could be, in parallel before or after the pathologist if we really want to provide prediction, especially if we are going to the extreme and we want to provide early detection, like when it's just started and like not at the low level, or as we call the outlier level, still in the beginning. That's very important. So that's one point. The other one, which clinical data are you, and how do you combine it? Because data mining is a big phrase. The question is, what are you really doing that? To combine clinical data, is it descriptive, is it physiology, whatever that you combine in order to define and to create the right prediction?
Yeah, actually, we don't really look at those images hypothesis-free. That's in principle not possible. You run into a combinatorial problem. If you look at everything that's possible in these images and have no clue what to look for, that's infinite. The possibilities are completely infinite. You have to have some hypotheses, and you don't have to pretend you are stupid. You know that there are nuclei which are important, that certain nuclei have a much bigger and maybe cancerous nuclei, that a healthy gland has a certain shape. You know that, and you put it in. And then for discovering new genes, you have to have also some hypotheses. For instance, that inflammatory cells might play a role. If you don't have this idea and you look for everything that's possible, it's simply too much.
I was referring to that. Once you choose your group and you have an assumption, then, you start for something. I'm not speaking about, Sure. You choose your images, and then you start, and you have to start to cluster them, to group them. I'm speaking on this level. And still there, you can go according to guidelines, and you can do it, to try to look on the low level or all levels, whatever you choose.
So the system will play with all these possibilities and comes to some conclusion. It might miss an important effect that could happen, but in the end, you certainly will have much more than you had in the beginning and what you have known before. You create this way new type of knowledge. You might not have grabbed the best one, but you at least made progress. Because if you have a combinatorial problem, you're never sure that you reach the absolutely maximum in your system. And you had a second question? Or?
No, regarding the combination with the clinical data.
Yeah, so the clinical
which kind of clinical data?
Is also very complicated. You need to know what kind of value you want to create, and then you have to look for the clinical data you need for this. And people at cancer centers, for instance, they collect all these clinical outcome data, but every cancer center does it a little differently, and it's not really standardized. That causes a big problem. But certainly what is extremely important is the recurrence of a cancer. That has a lot of information. Does the cancer recur after surgery or without surgery or after certain treatment? That's a very important data point which we use. And if you have additional information like how the patient was treated and what was the response to the treatment, if you have this information, it's even much better. And other types of data like age and things like that, we grab everything we get, but it's not really standardized in how the world collects data in this respect.
Neil?
Let me ask one question. Just on the context of the clinical validation of this, can you say something as to what data has already been tested or validated, relative to these discovering these maybe new spectrum on the pathology?
Yeah, right now we are in the state of collecting data, huge amount of data, but we just collected something in the order of 200 cases per organ or per certain case. So what we are doing right now, we're collecting thousands of data, but we have not worked on those data yet. So we are in the process of doing that, and at the end of the year, I think we will be much further, but it's work in progress right now.
One more question.
Rather a comment. So let's assume we take all the data we have now, with different qualities, whatever data we have. That means, and trying to classify them into subclasses somehow. That has to be a dynamic process, but you cannot re-evaluate all the data all the time because then the classification is gone. So that, but without evaluating it, it's not going to be helpful. So that's kind of a dynamic problem, I think, which has to undergo maybe a periodic quality statement re-evaluation of the data, and then it's more, and I think that's the way. It's really just the very beginning of a long process.
Yeah, that's perfectly true. I mean, the datafication has to be more or less standardized. If you have to reanalyze your data again and again, I mean, you don't have a valid database where you can look at again, have a new idea and test a new idea. It has to be more or less standardized. And if you bring in new data, you have to analyze those data with the same algorithms you have analyzed the old data. So that's what we have. One of our big project is not just to do image analysis, but just to do image analysis in a standardized way. And there we use actually also a lot of machine learning, where the system adapts automatically to the variation of the different images. They are very differently stained in the different locations, and even in one and the same location, they're sometimes stained very differently. Some overstaining, less staining, and so on. But through machine learning, we can solve this problem. And getting more and more, we are not there yet completely. So it's certainly a challenge here. We will never be there completely. I agree. Yeah. But you can do already a lot with the simple things. Yeah.
Sorry, we have to move on, but please catch Dr. Binnig in the break. Thank you very much, Dr. Binnig.